Once again to Biology 121 Online. This is video 3. Today we're going to be talking about photosynthesis, specifically the light-independent reactions, which involve carbon fixation and sugar production. Once again, just a quick review of where we've been so far. We've talked about photosynthesis as the opposite of respiration. Cellular respiration is the process by which mitochondria break down a sugar and release its energy in the form of ATP. Photosynthesis is just the opposite, where energy from solar radiation is trapped in the form of first electrons and then bound into ATP. That's steps 1, 2, and 3, which we've talked about previously. Go back and watch videos 1 and 2 for a review on those. And today we're going to talk about step 4, carbon fixation. This is the part of the process that happens even when it's dark, because we already have the ATP formed, and this is just the part where plants are going to take that ATP and store it in the form of sugar. So, skipping that, we're going to talk about the light-independent reactions in photosynthesis, which is where there's no longer a need for light. These actually, in some plants, happen after darkness falls. So the ATP might be stored for several hours, and then once it gets dark, we're going to form up that sugar from the energy that's in the ATP and NADPH. So remember that plants breathe in carbon dioxide, that's CO2, and they breathe out oxygen in the form of O2. So the only difference between those two molecules is the carbon, and that carbon stays inside the plant after the carbon fixation is called. That's why it's called carbon fixing. The word fixing in this context is similar to the word affixed, meaning like it's stuck in place. So the carbon is going to be drawn out of the carbon dioxide in the air and fixed into the plant, set in place. So here's our Calvin cycle, the carbon fixation cycle. Now this is a little bit like the Krebs cycle, but it runs in reverse. So just like in cellular respiration, it takes 2 cycles for every 1 sugar. So you can see we're going to start at the very top and work our way just around this cycle. So at the very top, we have these input of carbon dioxide molecules. We're going to have 3 carbon dioxide molecules per cycle, and that's the carbon fixation stage. Those carbon dioxide molecules are going to be stuck onto another molecule called rubisco. That rubisco then becomes a 3-carbon molecule. As you can see there, COO minus, CHOH, and CH2O with a phosphate group attached to it. There's 6 molecules of that 3-carbon molecule, 6 copies of that 3-carbon molecule involved in one single turn of this Calvin cycle. Then as we continue around the circle clockwise, you see that there's energy being used. We're going from ATP, the high energy version, down to ADP, the low energy version. So those ATP molecules have been charged up through the light-dependent reactions, and now they're being discharged, and the energy from them is being harvested and bound into this cycle. Same is true with the NADPH. Again, those high-energy electrons that were built up in photosystem 1 are now being stripped back out, and the energy there is being entered into this Calvin cycle. So that energy then is being used to form up bonds within a sugar molecule. So again, it says half a molecule down at the bottom, half a molecule of glucose. So that carbon is coming from the CO2, and there's hydrogen and oxygen that's coming from water, and that's our energy storage system. So now the energy from the ATP and NADPH that were charged up by the photosystems 1 and 2 is now going to be stored in the form of sugar, which is a very stable molecule if it is bound properly. So now that we've got our sugar out, the carbon has been accounted for, there's water that's been bound in as well. There's just a little bit left to do to finish this cycle. In order to finish the cycle, all we're going to do is says stage 3, regeneration of the starting molecule. So we've got this these molecules that are left over that are not ready to be rebound to carbon. So it takes a little bit more energy there over on the left-hand side, ATP being used up, in order to regenerate that rubisco and be ready for another crank of the wheel, as it were, for the cycle to continue on. So just like with the Krebs cycle or the TCA cycle, what we see is this circular process where, in this case, carbon dioxide at the top is being having the carbon stripped out. Energy is being used to form the carbons there into sugar molecules. and then a little bit more energy is needed in order to reset the sort of raw materials to bind more carbon dioxide and start the process over again. So now what are plants going to do with all of that sugar that they're producing? You may recall that there are two major polymers or long chains of sugar. All the way back to the very beginning of the year, back in September and October, we talked about cellulose and we talked about starch. And if you see, each one of those hexagonal rings is a sugar molecule. And they have been bound together at an oxygen through a dehydration reaction. Go back all the way in your notes to check that one out. And the difference between the cellulose and the starch is just an orientation difference. You can see that in both of those two, the first sugar is identical. It's got a CH2OH up at the top of the molecule. In the cellulose, the second molecule, the CH2OH, is down at the bottom. The third one is at the top. The fourth one is at the bottom. And in a starch molecule, it's got that CH2OH at the same position across the top, all the way across from one sugar to the next to the next. So in each of these cases, we're seeing 4 sugar molecules bound together. And the only difference between cellulose, which is the fiber that makes up wood, and starch, which is the fiber that makes up grain, which we can eat if we grind it up and cook it, the only difference is whether the sugar molecules rotate relative to each other or not. So cellulose is the woody parts of a plant. When a giant oak tree spends 120 years growing to be 100 feet tall, all of that growth is based on cellulose, which is really just made up of sugar molecules that the plant created using photosynthesis. All right, that's it for today. We're going to come back around on Wednesday and we're going to talk about some specific details in terms of different ways, different strategies that plants use in order to make sure that their light independent reactions are leaving the plant in the best condition for its particular environment. That one's going to be a little bit longer of a video and a little bit more complicated. So please do make sure you're creating some time in order to participate in engaging with that video on Wednesday.